Deoxyribonucleic acid. As we all know, DNA stores biological information and is the blueprint for life. In this video, I am going to demonstrate the extraction of DNA. Well, I didn't really figure this out on my own, but in 1869, Friedrich Mischer did so. The isolated DNA is used for research in the field of molecular biology and also for forensic analysis. I learned this experiment from a couple of YouTube videos and I wanted to try it today and share my experience with you all. Today I'm going to show you all the extraction of DNA from a strawberry. Strawberries are the perfect example for extraction of DNA for two reasons mainly. One, they yield a lot of DNA, and two, they are octoploid, meaning that they contain 8 copies of each type of DNA chromosome. These characteristics make the strawberry DNA easy to extract and also to see. So let me take you through the various steps involved in extracting DNA from strawberry. The three main components that help in the extraction are soap, salt, and isopropyl alcohol. To start with, pour distilled water into a beaker. Make sure you have taken 90 ml. The first component, the soap, helps to dissolve the cell membranes. Take 10 ml of soap and add it into the beaker. The second component, the salt, helps to break up protein chains that hold nucleic acid together, thus releasing the DNA strands. Add 1/4 teaspoon of salt to the solution. Stir it well, and now we have our very own homemade DNA extraction solution. Now place the strawberry into the Ziploc bag. Try extracting as much air from the bag as possible. Now squeeze it nicely. After squeezing, drop 4 tablespoons of the solution into the bag. Again, squeeze it up nicely. After squeezing, transfer the pulp onto a sieve and allow the thin solution to collect into the jar below. We now have our strawberry solution. Pour it gently into a test tube to about 50% of its capacity. We now move on to the third component, which is isopropyl alcohol. I have used surgical spirits which contains the same. Take 5 ml of isopropyl alcohol and add it to the solution. Now watch the test tube carefully. You will see a membrane forming in between the isopropyl alcohol and the strawberry solution. The DNA gets isolated as it is not soluble in isopropyl alcohol, especially when it is chilled. Pick up the membrane and place it on the slide. Now view it under the microscope. Wow, this is exciting. Let me now switch off the light and project it onto a wall. And this is what it looks like.